In this lecture, we're going to take a look at cellular reproduction and the process of mitosis. Cellular reproduction is important for 4 different reasons. First off, cellular reproduction can replace lost or damaged cells in an organism. It is also how an organism grows, and it's how organisms repair different parts of their bodies. And last but not least, cellular reproduction is how organisms reproduce sexually to make more offspring. So an overall picture of cellular reproduction looks like this. We're going to get 2 what we call daughter cells as the result, and they are going to be genetically identical, or genetically the same as the parent cell. In cellular reproduction, the cell has to duplicate its DNA-containing structures, which are called chromosomes, and these are what carry the genes of the organism, which will be later transmitted into proteins and have an effect on how the cell functions and how the organism looks. Now each cell also gets each cell is going to get an identical set of chromosomes, identical to each other and to the parent cell. Now there are a few different ways that organisms can reproduce themselves, like the whole entire organism themselves. Single-celled organisms obviously just divide. Okay, they can just go through cellular division and divide into an identical cell. And this is called acellular reproduction. There's some multicellular organisms that can reproduce asexually as well, like the sea star and the spider plant. However, most multicellular organisms need to go through what's called sexual reproduction, which requires the fertilization of an egg by sperm. And so the formation of sperm and egg involves a special kind of cell division, which is called meiosis, and that's what we're going to cover in the next lecture. So let's take a little deeper look at the DNA of the cell. That's where all the genetic information, that's where all of the blueprints of the cells and how it works and what it looks like is going to be stored. So a genome is a complete set of an organism's genes, and in humans we've got 35,000 genes. And these genes are located on the chromosomes, or the DNA, in the nucleus. Each chromosome is a very long DNA molecule that contains thousands and thousands of genes. In humans, we've got 46 chromosomes that package all the DNA. In dogs, there's 78 chromosomes, and in mice, there's 40. So chromosomes are made of chromatin. Okay, so there's some different names here that are very similar. We need to get kind of straight. So the chromosomes are made up of what's called chromatin, okay, which is DNA plus the proteins that are used to organize and help control the gene activity. So chromatin is DNA plus proteins, okay, and all of that wound together makes a chromosome. Now the proteins that are attached to the DNA and are helping control the this gene expression and holding it together are called what we call histones. So those are the special proteins that are associated with the DNA. And these histones are going to help package DNA into what look like beads on a string. And each bead is called a nucleosome. Okay, so each winding of a bead is basically a strand of DNA with 8 histones attached to it. All right, so and they keep winding itself up into these nucleosomes. These nucleosomes get beaded up on what looks like a string. We call that a chromosome. In fact, when cells aren't dividing, the chromosomes are very loose and kind of unwound. And so it's actually really hard to see the DNA in cells when the cell is not dividing. When the cell is dividing, we'll soon see that the chromosome or the DNA gets tightly wounded in the chromosomes. They get packaged really, really tightly so that the the DNA can be split into two and make two daughter cells. Okay, so before a cell goes through mitosis or cellular division, it needs to duplicate its chromosomes or duplicate its DNA. Otherwise, the two daughter cells will have half the amount of DNA that the parent had, and they won't be identical. So the DNA needs to be copied by what's called DNA replication, and we will definitely go over DNA replication later in future lectures. But that's basically how DNA gets replicated or copied. So each cell is going to have two copies of each of the chromosomes of the DNAs, and those are what we call sister chromatids. Okay, so if we see here, here's a chromosome, and then after it gets copied, it's got two copies in there. They're called sister chromatids, and in the middle, they are joined together by what's called a centromere. So it's like a little joint. That way, they know that the two copies are close together. They don't get mixed up in cell division, and they don't both go to the same cell, the same daughter cell. So when we talk about cellular reproduction and mitosis, we have to talk about what's called the cell cycle. It's basically just a sequence of events that goes from when the cell arises, okay, the new daughter cell is formed, to when it divides again. Now, most of the cell cycle is spent in what's called interphase, in which basically the cell is just kind of doing its normal day-to-day functions. So we can see that interphase basically takes up about 90% of the time of the cell cycle. And that's, the cell just hanging out doing what it's supposed to do. A lot of biologists call that, the time when the cell is just doing their normal functions, they usually call that G1 of the interphase. Okay, so that's the G1 part of the interphase. Because when the cell is going to kind of prepare itself to divide, it's still doing its functions, but it's kind of preparing, all the steps. And so we're going to get different subphases within interphase. Now, in order for the cell, the parent cell, to make two identical daughter cells, we not only need to double the DNA, but we also need to double everything else in the cytoplasm. We need to make, extra lysosomes and extra Golgi apparati and extra mitochondria as well. So during the cell cycle, the cell is going to grow and kind of double in size, making extra of all of the organelles inside of the cytoplasm, as well as duplicating the chromosome DNA. So when the cell is growing and doubling everything in it, it's what we call the S phase of the cell cycle. And that's seen right here. So the S phase is when the DNA synthesis and chromosome duplication occur, as well as duplicating everything else in the cytoplasm. Finally, just before the cell is going to divide, it goes through another growth spurt, which we call that the G2 phase. So the chromosomes are duplicated, they're wound up, they're ready to go. Everything is nice, the cell is nice and full and fat and ready to make two daughter cells from it. Now we've just talked about what happens in most of the cell cycle, which is interphase. We have the G1, S, and G2 kind of subphases within interphase where the cells are just doing their normal thing and or just kind of starting to prepare for cell division by duplicating the DNA and all of the organelles within it. The next part of the final part of the cell cycle is what we call the mitotic phase. This is about 10% of the time. So this is the part of the cell cycle where the cell is actually going to be dividing into two daughter cells, identical daughter cells. So there's two overlapping parts. One is called mitosis and the other cytokinesis. Mitosis is the division of the nucleus and the DNA, whereas cytokinesis is the division of the cytoplasm and all the organelles into two separate identical daughter cells. So here you can just write down exactly what I just said. So the mitosis part is where the chromosomes are distributed evenly to the two daughter cells, and then cytokinesis, divides the cytoplasm to actually form the two. It like pinches off the big cell to make two daughter cells. And interestingly, cytokinesis actually starts to begin before the mitosis is actually complete. So before all of the sister chromatids are separated into the daughter cells. So we get, starting the pinching off process before all of the sister chromatids are actually in the two separate sides. So in the mitotic process, the process of actually dividing the DNA into two identical daughter cells, there's actually 4 main stages. And these are kind of man-made artificial stages in a sense, because this is basically a continuous process. But you can look in the microscope at different cells dividing and see these different stages, within cell division, and kind of say, okay, here's where they're all lined up, here's where they're separating, and stuff like that. So these main stages are what we call prophase, metaphase, anaphase, and telophase. And although my name is not Patty, like my biology professor, I I remember the acronym Patty made a taco, okay, to help me remember the sequence of the mitosis stages. So prophase, metaphase, anaphase, telophase. Patty made a taco. So as we ready the cell for mitosis, it's going to go through that interphase, remember, which was 90% of the cell cycle. Well, towards, in the G2 part of interphase, we are going to, start to have that duplicating of everything. And in the cytoplasm, we're also going to get a formation of 2 centrosomes, okay, or a centriole pair. And these guys are going to be very important in pulling the DNA into 2 separate cells. Also, inside the nucleus, the chromosomes are duplicated, okay, but they're still kind of loosely packed, so we can't totally, distinguish each chromosome from each other. And then we still have the nuclear membrane. Now, this is going to start to change when we get to prophase. See, in early prophase, the chromatin starts to coil up very, very tightly, so that it can be thick enough for us to see with a microscope. And these chromosomes are going to again have 2 identical sister chromatids that are attached at a centromere. During early prophase, the nucleoli or the nucleolus is going to start to disappear. The nucleoli is where the ribosomal RNA is produced that is going to, make copies of the DNA and take it out to make proteins within the cytoplasm. But we'll get there in a future lecture. Now, in the cytoplasm itself during early prophase, we're going to have these spindles forming from the centromeres. So little microtubule proteins are going to start growing from the centromeres, and that's what we call the mitotic spindle. And you can kind of see that in right here. Okay, so these are the centromeres, and then going out from them, you've got these kind of finger-like things that are what we call the mitotic spindles forming. Now, as prophase goes on, later steps of the prophase, the nuclear membrane is going to start to break apart so that the DNA can be, just out in the cell and be able to be pulled apart. These microtubule spindles are going to start to attach themselves to the chromosomes at the centromere, at that center dot where the 2 sister chromatids are attached to each other. And the spindles are going to start moving towards the opposite sides of the cell, because we need one spindle on one side and the other spindle on the other so that they can pull those sister chromatids apart into 2 different daughter cells. All right, so in metaphase, the next phase of mitosis, those mitotic spindles are fully formed and the chromosomes start to line up kind of in the middle of the cell. So another way I can distinguish metaphase from the others, or with what's happening inside the cell, is I think of metaphase being, in middle, both having M. So the chromosomes are meeting in the middle at metaphase. The centromeres of all the chromosomes are lined up along the plate, so that means those little dots that are connecting the sister chromatids are all lined up in the middle there, and we get the microtubule spindles attaching to each of those chromosomes on the centromere. Now during anaphase, the sister chromatids are going to start to separate, start to be pulled apart. So that's how I remember anaphase. I remember A for apart. Okay, so those sister chromatids are starting to be pulled apart into the two poles of the cell, the two sides of the cell. So each daughter chromosome is going to migrate to the opposite ends of the cell during anaphase. And finally, during the last kind of the 4 stages of mitosis, which we call telophase, telophase is going to begin when the sister chromatids are fully on the opposite ends of the cell. And so we get a reverse of prophase, where we get the nuclear membrane starting to reappear. So the nuclear membrane is starting to be rebuilt around the chromosomes. Okay, and the chromosomes are going to start to uncoil a little bit, and we're going to get the reproduction of our nucleoli, so we can get RNA, and these little spindles are going to start to break down and disappear. And we usually keep cytokinesis kind of for last, because that's basically the pinching off of the big mother cell into 2 daughter cells. And although it's kind of happening from the start of mitosis, we always kind of keep it for last in the sequence, because that's kind of the last end all thing to make 2 identical daughter cells. So cytokinesis is the division of the cytoplasm. It occurs along with telophase. We get this little cleavage furrow, right there in the middle between, the 2 daughter cells starting to pinch off. Okay, and then these proteins are kind of wrapped around and kind of pinch off the 2 cells, the 1 cell to make 2 daughter cells. So there we have the basic process of cellular reproduction and mitosis. Now plants go through all the same types of steps. The only difference is that the cytokinesis phase. Okay, so they don't get a little cleavage further, pinching off of 1 cell into 2 new cells. What happens instead, because remember, plants have cell walls, so what will happen is after telophase, the plant cell begins to grow what are cell plates, like little tiny specks of the cell wall that just keep kind of growing and growing and growing together until it just literally divides the 2 daughter cells from each other. Ideally, normal cells will go through nice controlled cell division, where the cells have a cell cycle control system that will shut it down if something goes wrong. Well, if this control system is broken or not in place, malfunctions can occur that can lead to cells reproducing either at the wrong time or in the wrong place. And so that is going to lead to cancer. Now, there's many different types of cancer. We can have just a benign tumor, which is abnormally growing cells, but they remain in the site of development. They don't break off and start spreading all around. So I guess in a sense this is the best kind of cancer to have, because the cells just stay there. They're not spreading at all. But they still can cause problems, especially in the brain. Those cells can start dividing and kind of push on, healthy cells and, adversely affect the cellular communication inside of the brain, and so you get, some brain malfunctions. Now, what we think of cancer, not just like a tumor, but cancer, is basically severely deranged cell cycle control system. Like it's out of whack. There's no check system, and these cancer cells are, they are continually growing and growing and growing and dividing and dividing and dividing, and these guys have the ability to spread to other parts of the body. Okay, and that's what we call metastasis, is when a cancer has what we call metastasized, it's then spread to different parts of the body, and that is definitely dangerous. Cancer that is spread to other parts can quickly affect healthy tissues and, kind of push the healthy cells out of the way and cause lots and lots of problems. So cancer is usually named according to where it originated in the body. So a carcinoma type cancer is a cancer that's originated either in an internal or external covering of a body or a body organ. Sarcomas usually start in support tissues like muscles and bones. And then leukemias or lymphomas are going to be starting in the blood or blood-forming tissues, the lymph nodes and stuff like that. So medicine has come up with some ways to try to treat cancer. There's radiation therapy, okay, and chemotherapy. Those are the big, two avenues of cancer treatments. Radiation is, higher energy radiation waves being pointed at, where most of the cancer is, and those radiation waves can kill dividing cells, although it has side effects because it also kills healthy cells as well. Chemotherapy is usually a drug that's either ingested or injected into the veins that kills dividing cells. And again, chemotherapy drugs are not site-specific, so they'll go all over your body killing cells that are dividing. So again, there's lots and lots of side effects. We can also take out, some cancers surgically, which is good. If the surgeon can remove all of the cancer, then hey, we're cancer-free. But even if one little cancer cell is left behind, we can have, that cancer reappear. And obviously with any surgery, there can be side effects, infections, and stuff like that. So cancer is, as many people probably know, because it is prevalent in, our society and country that we live in, that cancer is not a fun thing to go through. So, but we do have some treatments that people can take as long as they understand the side effects. Sometimes they work, sometimes they don't. You just never know.